I'm Faith.
Hi, I'm Elshaddai.
Hi, I'm Anchal. And we are members of Relay for Life. Relay for Life is the college chapter of American Cancer Society, and as part of the mission delivery team, we hope to spread awareness about cancer, not only by organizing fundraisers or by forming great relationships with cancer victims, but also by educating people about cancer research. And we are here to interview Professor Coller, who is a professor in the molecular biology department, but also involved in cancer research.
In my laboratory, we're very interested in what happens when cells are not actively dividing and making a new cell. So lots of cancer research focuses on cells that are dividing and making new cells, but we're really interested in what it means to be a not dividing cell at any time. And we think that this is really important for cancer research because a lot of different types of tumors show something that people call tumor dormancy. So you have an initial tumor, and then sometimes many years later, the patient will have a recurrence of the tumor. And we're very interested in knowing what's happening to the cells during that period of time when they're dormant, when they're not actively dividing, and what it is that makes them become dormant and what it is that makes them become a tumor again. And so our research is focused on trying to figure out, could we keep the dormant cells dormant? Alternatively, could we take cells that are dormant, make them start to proliferate, and then kill them? And then third of all, can we design strategies to kill dormant cells specifically? When I was a graduate student, I got very excited about cancer research. And when I was a graduate student, I was very interested in understanding if there were chemicals in the environment that were causing mutations that were giving rise to people's cancer. And so we spent a lot of time looking at the pattern of mutations caused by different chemicals in the environment and the pattern of mutations that are actually in people to try to see if we could find the most important mutagens and carcinogens for people. So that's what I got excited to study initially. So I think that some of the major breakthroughs in the last couple of years in cancer research. So first of all, we've had the ability to sequence tumors. And so with the rapidly increasing capacity for sequencing DNA. A lot of people have applied that to understanding what the mutations are in tumors, and this has been very exciting. So one particular find was that there are mutations in two enzymes that people thought of as metabolic enzymes, maybe not so interesting, but actually specific mutations in those enzymes turned out to be really commonly found in certain types of cancer, and this has led to a whole new field of trying to understand what those mutations are and whether we can specifically target people with those mutations. Another big change that I think has happened in cancer in the last couple of years is the realization that cancer cells, they're not only dividing inappropriately, they're also growing. Because you can imagine if you just had a cell that kept dividing, it would keep dividing, you'd have lots of cells, but they'd be very small. So actually cancer cells not only divide, but they also get big. And actually that cancer is associated with a reprogramming of metabolism along with kind of inappropriate proliferation. And a lot of the genes that we thought of as oncogenes and thought of in the context of causing cells to proliferate actually also help cells to take up nutrients from their environment and use them to make more proteins and lipids and carbohydrates that the cell needs to bulk up. I think the biggest challenge facing the field now is to manage all of this sequencing information. So I hear from patients and patients' families that even if their parents or their loved ones tumors have been sequenced, and even once we know the mutations, it's still not clear how to personalize the therapy. And so I think that's kind of the biggest kind of challenge for the field right now, is to go from that DNA sequence to a really effective cure for each patient. Oh, all the time. We have many setbacks. I mean, when you do research, every day there's all sorts of issues that we need to address. Today all the cells are contaminated. The next day there are Western blots that don't reproduce. Doing research is always about kind of dealing with challenges. If you're kind of at the cutting edge of anything, you're always having setbacks, always realizing that something that you thought was true isn't true, that you've misinterpreted something, that you should be thinking about something differently. But we try to deal with all of them in the same way, which is first of all to be calm, and the second of all to try to be data driven, to, we develop models of how we think the cells are working, how we think cancer cells are working, how we think that other cells were working. And then as we generate more data, we try to think about whether it fits our model. And if our model doesn't work, then we try to come up with a new model. So our main goal is to try to use the data to help us to develop our models and to not be so in love with our models that we don't give them up. There's a lot known about kind of the cycle-dependent kinases that are really important regulators of the cell cycle. And so we have a good understanding of that. What's less well understood is kind of what happens to cells when they exit the proliferative cell cycle. So that's part of the reason why we're really interested in that, is because we think that there are important things for cells to do when they're not cycling, but there just hasn't been as much attention given to that. I don't think it's, it is kind of an exciting and challenging thing to study, in part because we don't have, for us, the challenge is that we don't have great markers. So we don't have great markers for what it means to be a quiescent cell, and so we haven't been able to kind of pull the quiescent population out from people. And so one of the things that we're working on in this laboratory is finding better markers for the quiescent cells so that we can identify them and ask what's special about them, what's making them tick, and what's regulating them.
Sorry, can you explain what quiescent is? Just in case people don't know.
Sure. So quiescent is what I said at the beginning. So cells can be proliferating, making new cells. Mm-hmm. When cells enter a quiescent state, it means that they're not actively dividing. So cells kind of have a choice: should I divide or should I not divide? If they choose not to divide, then they can be quiescent. The thing is, cells that aren't dividing can be quiescent, or they can be terminally differentiated, they can be senescent, they can be dead. But quiescent ones are the ones that we're very interested in because they're the ones that are not actively dividing, but they have the capacity to start to divide. I think that one of the things that, it's nice to think about inventions. I think that two fields that I'd love to see more progress on. One is things that target things to tumors. So a lot of the times we maybe have a great idea for how to treat the tumor, but it's really hard to get it to the tumor. And there's some very exciting work on kind of tumor targeting. The other thing is that I think is a little bit underappreciated maybe are the mouse models. So mouse models of cancer are really powerful approach for trying to understand human disease before you're ready to start treating patients with something which, of course, involves actually involving people. And so the mouse models of cancer, I think, is a very exciting field. There's been a lot of wonderful research and a lot of advances. As just one example, a mouse model was just published that we're excited about taking advantage of that kind of does simulate the dormancy. And so they managed to have a full-blown tumor, then there are micrometastases that take several months, and then those develop into organ-specific full-blown tumors. And so more mouse models so that we can ask, okay, what's special about those cells? What are they doing? And do these treatments actually affect it? I think would be really beneficial for the entire field of cancer research. I think that for a while we focused a lot on proliferating versus quiescent fibroblasts, and one of the changes for us has been we'd like to take what we've learned about proliferating and quiescent fibroblasts, which has been much of the focus, and translate it more directly to cancer research. So like right now in my laboratory we have a medical doctor who's from Israel who came to do research with us, but she's actually treated cancer patients, and we brought her here in part because she would have kind of a clinical perspective to offer to us. So that's been one of the kind of changes in my lab over the course of the last couple of years, is more of a focus like directly on cancer research. Right. So I think, for myself, I think that there's a really important role to play in cancer research of basic biology. I think that, we still have the opportunity to learn really fundamental things about cancer in the laboratory, and that some of these will turn out to be translatable in ways that aren't really obvious from the outset. So for instance, Heather Christoph, working with Luke Hantley, discovered that there's a specific enzyme in the metabolic pathway that's only expressed in cancer cells. That's actually the splice variant, just one exon comes in or out, and actually one particular splice variant is only expressed in cancer cells, not the rest of your body, and in embryonic cells, so it's kind of re-expressed. And people are now realizing that the special ways in which that particular enzyme are regulated are actually important for tumor growth. And if you make small molecules that force that enzyme to be in a specific conformation, you can actually inhibit tumor growth. And so those experiments were started as very basic research, but now there are companies that are built around this, doing research that looks like it might have the potential to help patients. So that's one thing that I think is really important. I also think that another field that I think is really exciting that's emerging is the field of immunotherapy. So there's been a lot of advances in using a patient's own immune system, kind of engineering their T cells to specifically attack the tumor and giving those T cells back to the patient.